And so we've been talking quite a bit about sleep in this conference, but given the fact that we've been spending also quite a bit of time sleeping throughout all our lives and we still don't understand why, I think that's kind of warranted. And so I wanted to maybe talk about something related and present also something related to sleep. So when we're awake or when we're asleep, obviously the difference for us is pretty obvious, but also the difference in terms of global macroscopic brain signals is pretty clear. As Alain was showing, if you look at the electroencephalogram, for example, you have signals in awake brains that are very small and complex. You could say that they're low energy and high entropy. But then when you're sleeping, you have signals that are large amplitude and simple. So you could say that they're high energy in terms of the signal energy and low entropy. And actually, Jennifer is going to talk a lot more about those macroscopic measures. And what I'm interested in this talk is how do these dynamics emerge from microscopic interactions? How do the neurons talk to each other such that such dynamics can emerge? And so, how can we study this? Well, there's this device that looks a bit scary, and that's called the Utah array that can be implanted inside the brains of human subjects or human epileptic patients or of monkeys. So for patients, it's like implemented far away enough from the epileptic focus, and then you can record really hours of data, periods of wakefulness, periods of sleep, and hundreds of neurons. And you can also look at the spike waveforms and classify neurons as excitatory or inhibitory, regular spiking or fast spiking. But then when you look at this data, it's kind of a mess. Like you can't really make sense if it's not as obvious as with the macroscopic data what is going on. So how can we figure out what's going on in data that looks very messy? Like we look at pairwise covariances, or like Daniele was saying, like maybe pairwise interactions are not enough and we need higher order statistics. And so how can we address that question? Well, are pairwise interactions sufficient, for example? And how we can look at this is that we could have a null model for pairwise covariances. So we could have the most generic model that would only reproduce by design the mean firing rates and the pairwise covariances between neurons in the data and nothing else. That means that it assumes maximum entropy on all of the firing patterns of all the neurons, while only keeping these two statistics from the data by design. And if we write down this model, it takes this kind of form where each neuron's firing rate depends on its own bias towards firing, as well as this coupling parameter, which will be, which when it's larger will mean that these two neurons will be more likely to fire together. And now we have this model, and we can fit all these parameters to be able to match the statistics, so to match pairwise covariances from the data exactly. But now what does it mean if it's a good model? If it's a good model, it can reproduce other empirical statistics, other statistics than the ones that it was designed to reproduce. And that's exactly what we test here. We're looking at the probability that K neurons are firing at the same time in the same time bin. And when we look at this, we can see that the data is actually pretty well fitted by the model, which is the dashed line here, in wakefulness. But then in sleep, you can see that actually there's a little bit of a difference. Like it's a bit, sometimes the model is higher, sometimes the data is higher. So maybe the pairwise correlations, pairwise interactions are not enough for sleep. And if you look actually in more detail, you can see that for sleep, it's in particular for inhibitory neurons that the model is failing. And so especially for inhibitory neurons, maybe higher order interactions are needed. And so how do we test this? Well, we can do the same thing, but this time look at maybe not interactions between each of the pairs of neurons, but each neuron and the population as a whole. So the highest order in a way that you could possibly look at. And so how do we do this? When we can build a maximum entropy model that would reproduce the probability that this neuron is active, given that k neurons in total in the population are active in the same time bin. And so will this model work better during sleep? Will it work better for inhibitory neurons? And how do we find out? Well, we can look, as before, at other statistics that this model wasn't designed to reproduce. We can look at, for example, pairwise covariances. And that's exactly what we do. And so here we can see pairwise covariances in the data as well as in the model. And we can see that in both cases, the model works pretty well, but it works quite a lot better for sleep, which shows that these neuron to population interactions are more important in sleep, and especially so for the neurons that are in red here, for the inhibitory neurons that are particularly more correlated. But we're still not quite so happy here, because for pairs of inhibitory neurons, we can see that the model tends to underestimate how correlated they are. So is there something about inhibitory neurons? Do they interact more like between themselves rather than with the population as a whole? And so how do we study this? Well, we can refine this model to take into account separately the interactions between each neuron and the excitatory population, and each neuron and the inhibitory population. And so when we do this, we expect to maybe have a better model for sleep. And that's indeed what we see here, that the model is performing better now at fitting the pairwise correlations, and also stops underestimating them. But you might see here we change from a model with less parameters to a model with more parameters. So maybe that's just why we're doing better. Is that really why, or does it really matter which populations we chose, which are the identities of the excitatory and the inhibitory neurons? And so to study this, we can shuffle around the labels of the neurons. So change which ones are excitatory, which ones are inhibitory, which within each of these subpopulations, and see how much the performance gets worse. And here the performance at modeling the pairwise covariances is measured by the mean square error. And you can see that it gets a bit worse, significantly worse in the human, and it also gets a lot worse in the monkey, which shows that these cell types, these excitatory and inhibitory types, are really important. And since we saw that these inhibitory cells are more correlated than, especially correlated within among themselves, maybe they play an important role in organizing these population-wide dynamics in sleep. Like Mavi was mentioning, like inhibition maybe plays an important role in the fact that we have synchrony, the fact that we have slow waves. And so in wakefulness, we can basically measure and we can basically have pairwise interactions that are a pretty good measure and can explain the kind of activity that we have. And these can help the emergence of these highly complex and asynchronous dynamics, so high entropy and low energy. And in sleep, we need more high order interactions with each neuron interacting with a cluster of its own cell type, and maybe inhibitory neurons playing an important role in organizing synchrony, organizing the slow wave patterns. And so when we stumbled upon this cluster kind of model, we thought maybe, like, is there any relevance of that? Like, why do neurons behave like that? Like, why would they have these high order interactions? Like, does it help at all for neural coding, for when we see a stimulus, how our brains encode it and eventually respond to it? And in the context of neural coding, it's important to think that each neuron doesn't see the stimulus as a whole, but rather sees only information about the stimulus from the neurons that are presynaptic to it. And so it might be important to look at populations of synaptically connected neurons when studying neural coding. But most of the studies that have been done in the field have studied random samples of neurons across the cortex. And so how do we study the two? How do they change statistically among themselves, and how do we approach this? Well, this is recordings that we have from our collaborators from the lab of Botan Roska in Basel, where they recorded in the layer 4 of the visual cortex of the mouse. And they used rabies tracing, which allowed to record, to infect neurons that were all presynaptic to the same neuron and record the activity of these neurons. And then we compared that to just neurons sampled randomly in the brain using also calcium imaging. And so when you look at the activity of these neurons, you can look at the fluorescence and how it varies throughout time. And we thresholded that to see when different neurons were active or silent within the same time bin. And so when we do that, we can start already doing simple statistics. We can start looking at the mean and the variance of this. And if you do the standard deviation divided by the mean, you can get the coefficient of variation. So that's what we started with. And we can see here that the coefficient of variation is actually larger in presynaptic recordings than in randomly sampled neurons. So maybe these neurons, the presynaptic ones, are more active and silent together. They vary more coherently. And that's especially true for between 2 and 22 neurons being sampled. So maybe we have clusters in the population that are about that size, where the population is about up to hundreds of neurons. And then we wanted to look in more details, like, are these neurons really more correlated? And so we looked at pairwise correlations between neurons, so just how likely they are to be active together in the same time bin. And here we find that if we look at different mice, there's not so much of a systematic difference between presynaptic and random. So maybe pairwise correlations are not the best metric for this. Like, then what is a good metric? Are pairwise correlations important or interesting to describe this kind of system? And as we saw before, we have the pairwise model that has been shown to be a great model actually in many recordings of randomly sampled neurons, except sleep, as we figured out, but still many recordings of many different species and brain regions, but always randomly sampled neurons. But now for presynaptic neurons, what would be a good model? And since we saw that they were kind of varying coherently and maybe also organized in these clusters of 20-ish cells, we thought of applying the cluster model that we introduced before, and to use to cluster neurons based on their activity, and then apply this model and see how well it will reproduce statistics. And so to compare these models, we want to see what statistics they're not designed to reproduce, what can they reproduce. We think that the pairwise model could be a good model for randomly sampled neurons, but what about presynaptic? And to find a statistic that they can both produce, we looked at the probability of generating different spiking patterns and compared that to the data, and normalized by the number of parameters. And that gives the Bayesian information criterion. And we can see here that for the randomly sampled neurons, the pairwise model is the one that does better. So this ratio is larger than 1. So the pairwise correlations seem to be dominant there. But then if we look at presynaptic neurons, this isn't quite the case. So maybe higher order interactions are needed. And how do we look at that? Well, we can do the same as before. We can look at pairwise correlations and how well this model is capturing them. And we can see that this model is capturing them better, as in the correlation between empirical and model predicted correlations are more similar in the in presynaptic neurons. But then we can ask the same question: do the clusters that we made, like, do these really matter? Like, does shuffling the labels really change the performance? And that's what we're doing here. We're shuffling the labels and we're looking at how much the error is increasing. And when we do this, we can see that for random neurons, if you shuffle the labels, the error gets a bit worse, it doubles. But then for presynaptic neurons, you can see that it gets much, much worse. It's multiplied by 5, 6, 7 times. And so this difference means that the clusters that we have in presynaptic neurons are a lot more important, and they're really dominant in structuring the spontaneous activity that's going on in these neurons. And that probably cannot be accounted by cell types, because cell types like also exist in random neurons. And as we saw before, the difference, like the worsening with shuffling, wasn't that big with just excitatory and inhibitory cell types. So that was for responses to uniformly gray stimulus. But now, how does this affect the way these neurons encode information, the way they respond to different stimuli? And so we looked at this by presenting different gratings with different orientations and different speeds. And we thought, our neurons are in the same cluster, are they going to be more correlated than any other neurons? And so when we look at this, we can see that neurons in random populations, if we look at them in the same cluster, we look at their Pearson correlation in terms of their mean responses to stimuli averaged over time in trials, and we divide them by shuffles over clusters. That gives a baseline for the whole population. And you can see here that this ratio is larger than 1. So neurons are in the same cluster show more similar responses to stimuli than neurons that are in different clusters, where the clusters were defined from their spontaneous activity. But now, when we look at the presynaptic neurons, surprisingly, this isn't quite the case. So these neurons are less correlated than you would expect by chance, um, as given by random neurons. So neurons that are in the same cluster are less similar. So the correlation structure that was really dominated by the cluster structure between these presynaptic neurons is changing more between when you don't have a stimulus, or when you have a uniformly gray stimulus, versus when you have these moving stimuli. So maybe the correlation structure and the cluster structure is more sensitive to the presence of a stimulus for presynaptic neurons, which we know is important for these neurons to actually encode the stimulus for the postsynaptic to read out and to get relevant information. And so in this project, we concluded that, again, high order interactions are important. Like in presynaptic neurons, you really need these high order interactions, and you can understand spontaneous activity as being organized in clusters, even though you didn't really need these high order interactions for randomly sampled neurons. But then for responses to stimuli, the fact that you have these clusters maybe allows presynaptic neurons to change their cluster structure and to be more sensitive to stimuli by changing their cluster structure and their correlation structure. And so to conclude on both of these stories about maximum entropy applied to different contexts. So we used methods from maximum entropy models that are from statistical physics, information theory, and we used different constraints, and they helped us reveal that correlation structures was different in between different recordings, and in particular pairwise interactions and pairwise correlations are not always the most important thing in data. Sometimes these cluster models are a more interesting way to look at the data, or a more relevant way. And we saw that the microscopic correlation structure is changing between brain states. So how these high energy, low entropy, etc. trade-offs are going on emerges from how neurons talk to each other at the microscopic scale. So that's maybe a link, like how we can use energy and entropy metrics at the microscopic scale to explain things that Jennifer is probably going to explain, to show more about at the macroscopic scale. And also finally the cluster-wide correlations that we see in presynaptic neurons might be helpful to encode stimuli. Because you imagine that if you have a code that's very high entropy and low energy, like the neurons are very desynchronized with very complex low order correlation patterns, that would give you a high dimensional code, a code that can code a lot of different things. But it would be hard to read out and sample. And if you go to the other extreme, if you have a code where the neurons are all tuned to the whole population, so they're very high energy, synchronous, but also low entropy, then that would be easy to read out. It's very robust. If one neuron's like active, but then when it should be silent, but then the other one's the opposite, you might still be able to encode that there's a cat and you should run away if you're a mouse. And so you would have a code that's more robust and easy to read out, but it would be lower capacity. Because all the neurons are basically very redundant. And so maybe the fact that you have clusters is a trade-off between having a high energy and high entropy code, and between robustness that's given by energy and synchrony, and complexity that is given by essentially entropy. So thanks a lot for your attention. And yeah, I'd like to thank my collaborators, and in a picture of a cat, because cats are very a maximum entropy animals. And I'm graduating in the fall, so I'm looking for a postdoc, and please contact me. And I'm open for any questions, of course.